hats. One is as the Editor-in-Chief of Journal of Vaccines and Vaccinology, which is one of the sponsors of this meeting, and I'll give a plug that we're inviting all of the presenters for this meeting to publish a paper in a special supplement of the journal. We've negotiated a major discount on the article publication fees from Omics. So I think it's only $400 instead of the normal article publication fee of $900 to submit a paper to the special edition. They'll be reviewed just like a normal paper. But as I say, I'd encourage all of the speakers and poster presenters at the meeting to consider submitting a paper. The other hats I wear, I'm an active clinician and I'm an academic with a university, and I'm also the founder of a company, Vaccine, which works in the area of vaccine adjuvants, and that's what I'm going to talk to you about today. So we've heard a very nice speech from Ray this morning about the problems that we face, or the challenges in the vaccine world, and I'm going to sort of dig down a bit further away from the politics and more into the science of what are the scientific challenges. And I guess this encompasses at least some of them. As we've heard already, we have some successful vaccines, and then we have some partially effective vaccines, such as the influenza vaccine, where we know we only get about 50% protection at best, depending on season. And then we have the other vaccines, like the cancer vaccines, where we've got close to 100% failure rate. So we know we can make successful vaccines, but the question is, how do we take some of those other vaccines and improve their effectiveness? As well as that, I work a lot in the pandemic arena, and there the major limitation is not so much designing a successful vaccine as making enough of it quickly enough to have an impact on an emerging pandemic. And so there we need to find ways to make our antigens faster and also more efficient, so we can use less antigen per vaccine dose. We've already heard this morning about the heterogeneity of cancers, and that's also true of infectious diseases. I think it's particularly evident in influenza, but we also have that an even greater problem in the heterogeneity of HIV, where each person is infected with thousands, if not millions, of different serotypes of HIV. Which is significant. In Ebola, for instance, we have the similar problem. We have Ebola emerging and adapting, and it's no longer a singular serotype that we're necessarily dealing with. So making vaccines that are protective against a specific genotype is not good enough. We need to find a way to get broader heterologous protection. Another thing that's often not talked about is that a lot of the people who need vaccines are actually the people who respond worse. And I guess the example is a cancer patient who has severe cancer. Not surprisingly, their immune system's pretty shot. And so although a vaccine may be effective against cancer in someone with subclinical disease, unfortunately the person with the most severe disease and the biggest problem is going to be the hardest one to actually get to respond to the vaccine. So we need to have strategies to improve vaccine effectiveness in poor responders. So that's very young babies, again, a very high-risk group for infectious disease. The elderly, another high-risk group, people on immunosuppressive drugs, who unfortunately are all the ones who are most susceptible to mortality, whether it's from infection or cancer. They're the hardest ones to actually protect. Again, in the context of pandemics, we want to give faster protection. It's no good if your vaccine takes a few months to take effect if you're going to die tomorrow. So you really not just need to give people vaccines quickly, but you need the vaccines to work very quickly. And alongside that, we ideally, particularly in a pandemic, we'd like to protect people with just one shot, so they have it and they know they're protected. We also want vaccines that work for longer, because humans, unfortunately, unlike mice, can live for 80 to 90 years. So it might be possible to give a mouse a course of vaccine and protect it for its 1 year lifespan. It's not so easy to do that in a human, where you want to protect someone potentially for 80, 85 years, and you don't want to be having to vaccinate them, every few months or every year for the whole of their lifetime. And other desires, I guess, particularly of the Gates Foundation, they're very keen on getting rid of needles in vaccines because there are risks and costs associated with needles, and also to get rid of the cold chain because this is very expensive in delivering vaccines to the developing world. So how do we go about, I guess, tackling these challenges? Well, I guess my job is to convince you that the best way to tackle most of these, alongside improving the quality of our antigens, is to work on the immunological side of the vaccine, which is to incorporate an appropriate adjuvant. Now, adjuvants sort of get either a very positive spin or a very negative spin, and that's why I entitled my talk, Adjuvants, the good, the bad and the ugly. There are those who love adjuvants, and equally there are a lot out there in the community who hate adjuvants. And so I guess I wanted to really tackle some of the scientific issues and say, well, adjuvants aren't bad and they aren't good. I mean, it really comes down to how you use them. And if you use them appropriately, then you'll get the benefits without the downsides. Again, my job is not to create bias by convincing you there's any one solution. This just alerts to the fact that there are a number of adjuvants out there. At the moment in humans, in fact, aluminium salts still represent 99% or more of vaccine use. So for 80 years we've relied on alum, and we continue to do so. There have recently been some newer approvals in the last 10 or 20 years in Europe of oil emulsion adjuvants in influenza vaccines. Also in the papillomavirus vaccine, there's now a combined adjuvant with monophospholipid A and alum, and essentially that's it. So there's lots of talk about adjuvants, but there actually isn't much happening in terms of products that are being delivered with novel adjuvants. So about 15 years ago, I chanced upon an elderly scientist who convinced me of the wonders of invention. So Peter Cooper had been playing essentially with this sugar. So this is inulin. It's a polymer of fructose with a terminal glucose. So essentially it's a modified form of table sugar and honey put into a linear array. And inulin's been used for about 60 years for kidney function testing in humans. So you inject about 40 grams of inulin intravenously to a human, and because it's inert, it just comes out through the kidney. So we know it's a very safe sugar. The only difference is this is completely inert. So the inulin they use for kidney function testing is soluble. What Peter had found is that if you just take this inulin and you now crystallise it into these, they look like little donuts, about 1 micron in diameter, and you mix it with an antigen and give it to an individual, you get this beautiful enhancement of the adaptive immune response. And I think for a long time people really just didn't believe this could be true. Fortunately, we now have a lot of independent data to show you that it is true. This is perhaps the most convincing. This was a study done by the US government, so it was completely independent, looking at avian influenza, which, as we know, in humans it causes about a 60% mortality, so it's a serious threat. Fortunately, it hasn't yet learned how to spread human to human, but obviously highly pathogenic if it did so. And so the US government, in partnership with Adventis, had developed a vaccine which the FDA approved. The only problem with this vaccine is it doesn't work very well. So if you take the FDA-approved vaccine, you immunise ferrets with 2 doses, even at a 22.5 microgram dose, and then you come in and challenge them with a homologous challenge of H5N1 influenza, only about 2/3 of the ferrets survive. So a third die. That's with the FDA-approved vaccine. So you'll protect some people, you're not going to protect everyone. So how do we improve this? Well, the answer is if you just take this and add in our inulin adjuvant, then what happened is you get 100% survival of the ferrets. Not only do you get 100% survival, but in fact you can get 100% survival at a much lower antigen dose, and in fact you can get 100% survival with just one immunisation, rather than getting 2/3 survival with 2 immunisations of the approved vaccine. So it clearly is doing something. The question is what's it doing? Well, just to show you that it's not peculiar to influenza, this is some data, again, from a commercial collaborator in Europe who's developing a vaccine against herpes simplex virus. So in their murine model, they immunised with a recombinant antigen from HSV, and they combined it with a number of different adjuvants, so CpG, which is a TLR9, another proprietary adjuvant, so they weren't able to tell me the name of it, and our inulin adjuvant, which we call Advax, and then the controls. And you can see that when the animals that have received the control are challenged, they all die. If you look at the animals that received the protein with a CpG, which is quite a potent Th1 inducing adjuvant, you can see you get about 60% survival. If you formulate with this simple sugar adjuvant, you see 100% survival of the animals. Similarly, if we look at a heterologous challenge, so this is now asking if we move from a homologous to a heterologous challenge, do we see benefits of the adjuvant? So this is some data where we've immunised animals with an inactivated Japanese encephalitis virus antigen. And then, in fact, again, a US government-funded project in the US, they challenged the animals with West Nile virus, which is a related flavivirus that's a major problem in the US. And suffice to say, if you immunise the animals with Japanese encephalitis antigen with the inulin adjuvant, you get not just 100% protection against Japanese encephalitis, you now see 100% protection against West Nile. So in some way, it's not just improving the homologous protection, it's improving the heterologous protection. Again, as I said, the important thing, though, is that generally you're using healthy adult animals for these studies. This is just to show you that it also works in neonatal models, which are much harder to protect. So this is an influenza challenge model in neonatal mice, where we take an inactivated virus, we immunise them with various adjuvants. You can see here we've given the inulin adjuvant, a oil emulsion adjuvant and CpG, and then we've challenged the neonatal mice, in this case after 2 immunisations, but we can also show this after 1. And you can see the only mice that survive after challenge are in fact the animals that have received the influenza virus with the inulin adjuvant. And in fact, in this case, CpG doesn't protect. But in fact, in adult mice, CpG does protect. So it shows that neonates, for some reason, are unresponsive to a TLR9 agonist or an oil emulsion, but they remain responsive to this sugar adjuvant. One of the interesting questions we observed early on was the lack of reactogenicity of the adjuvant, which perhaps is not surprising because it is a simple sugar. And so people ask, well, how do you explain the lack of reactogenicity? And also, how is the adjuvant working? Is it working through a TLR pathway? Because given it's so effective, everyone presumes that the only thing that's effective in this world is a TLR agonist. So we took mice that in fact are knocked out for MyD88 and TRIF. So this knocks out all TLR signalling in the animals. We immunised them either without or with the adjuvant, and when we looked at antibody titers, clearly the adjuvant was working beautifully in the TLR knockout mice. They were getting marked enhancement of their antibody titers. And similarly, when we challenged them with an influenza virus, the animals that received the inulin adjuvanted vaccine survived, whereas as before, the animals that just received the inactivated virus alone didn't survive. So obviously its mechanism of action is completely TLR independent. Interestingly, we then asked, well, is it working on memory B cell responses or memory T cell responses? It turns out it works on both, because if we take mice that have no B cells, so these are mu-MT mice, and we do the same study, so we immunise them with the inulin adjuvanted antigen or just the antigen alone. We then challenge them with influenza, and what you can see is even in the absence of B cells and the absence of antibody, all the animals that have received the inulin adjuvanted vaccine all survive, whereas the ones that get the standard vaccine all die. So obviously it's able to protect not just through a B cell response, but in fact also in the absence of B cells through a T cell response. So again, I'm not going to go into it today, but essentially this has now been tested in many different models around the world, not just of standard inactivated viruses or proteins. It works with peptides. We, in the last year, through a commercial company, have shown it works with live viral vectors. There's some interesting work emerging in the field of cancer, Alzheimer's and immunotherapy, where it also seems to work, and it's been extensively tested across pretty well all the animal species we've been able to get our hands on, and it seems to work across species boundaries. And just to show you that it also works in this odd species, humans, which sometimes people say mice lie. The question is, do horses and camels and goats lie? Because we've also shown it works in them. And in fact, it works very nicely in humans. This is some data on the 2009 pandemic influenza vaccine study that was done with a recombinant hemagglutinin, either alone or with the inulin adjuvant. And you can see we had beautiful enhancement in humans of the anti-influenza response if they received the inulin adjuvant, so confirming it does work in humans. So just quickly then, because I guess the key question is, one, proving to you it works, which has taken about 15 years. As Alan Cripps, who was with me at the beginning, will attest, it's hard to convince people something really is too good to be true. And so the question then is, well, how does it do it? Well, I've shown you already it doesn't work through TLR pathways. I can also tell you it doesn't work through inflammasome pathways, because we've also tested it in NALP3 knockout mice, and again it works beautifully. So it doesn't seem to work through any innate immune pathways that we can characterise. The other thing we need to explain is the lack of reactogenicity. So for those of you who aren't familiar, this is looking at injection of PBS, looking at the histology at the injection site. Basically, you don't see much. Alum, you get some moderate inflammation. An oil emulsion, you get very severe inflammation. And not surprisingly, the reactogenicity of these formulations go up with the level of inflammation. So what we asked next was, well, if we're not getting any reactogenicity, to which some of us who've had the vaccine can attest, maybe there's no inflammation. And when we threw out this hypothesis, of course, immunologists said we were crazy, that the whole way an adjuvant works is through induction of inflammation. So how could you possibly have an adjuvant that's effective and is not inflammatory? It was sort of like an oxymoron. Well, it turns out that, in fact, the hypothesis is right. Just to show you, this is a nice study in camels done in Dubai, where they are having problems with vaccine reactogenicity. This is a draining granuloma at the injection site with an oil emulsion adjuvanted vaccine against African horse sickness. So they asked for some inulin adjuvant to test in the camels. Cut a long story short, they got no reactogenicity with the vaccine, so they were very happy compared to the reactogenicity of the typical veterinary vaccines. And when they looked at the immunologies, they looked at the antibody titers to African horse sickness against the reactogenicity scores, you can see that the only exception to this rule of more reactogenicity goes with more immunogenicity was in fact the inulin adjuvant. Very nicely immunogenic, no reactogenicity. To show you that isn't just an artefact of camels, This is a mouse study done, in fact, by a collaborator, Todd Merkel at the FDA. And so he got interested in using the inulin adjuvant with an anthrax vaccine, and he was able to do this study where he used an injected dye that gets cleaved by the presence of inflammation. So this is a ProSense reporter. So you can actually do in vivo imaging of the mice to look at how much inflammation your vaccine's induced. So he injected an anthrax vaccine with alum, which is actually itself not very inflammatory. And so this is looking at the fluorescence of the reporter, showing you that there's quite considerable inflammation associated with the injection of even the alum-based vaccine, and you can see dramatically reduced inflammation with inulin, even when it was combined with murabutide, which itself is an inflammatory agent. If you just gave inulin alone, you really see no inflammation. So it really is non-inflammatory. So how is it working? Well, again, we don't know everything about it. We know that it's enhancing antibody responses. We also know it beautifully enhances CD4 and CD8 memory T cells against the co-immunised antigen. It enhances all of the, memory cytokines, both Th1 and Th2. So you can see here, if you give influenza with the adjuvant, increased IL-2, IL-5, 6, gamma interferon, GM-CSF. Interestingly, though, not the inflammatory cytokines. So interleukin-1, you can see, in fact, it looks like it's reduced, although it's not significant. Similarly, tumour necrosis factor alpha. So what it is an anti-inflammatory adjuvant that's enhancing the adaptive immune response in both a Th1 and a Th2 direction. Again, how is it doing that? How is it inducing this beautiful gamma interferon response without inflammation? The answer is, unfortunately, I can't tell you yet because we simply don't know, but that's what it does. So I showed you before that it enhances the T cell protection in mice that don't have B cells. Conversely, we can show that we can transfer protection with the memory B cells that are induced in the presence of antigen. Again, this is in the context of the Japanese encephalitis heterologous protection against West Nile, and what was shown by our collaborators is they could transfer the protection by harvesting B cells from immunised mice at both 3 months and also at 6 months. So it's inducing a long-lived memory B cell population that transfers protection. In terms of the molecular aspects, which I'll just finish with, of protection, we know that if we label these inulin particles, which we can make them fluorescent, and then we just put them in a culture with human peripheral blood mononuclear cells, the mononuclear cells gobble these particles up like they're breakfast. And so what you can see, this is just after overnight incubation, you can see all of the sugar particles have been phagocytosed by the human monocytes. And then if we look at gene signalling within these monocytes in response to eating this inulin, they get a big energy boost. And interestingly, if we look at what happens to the inflammatory genes, so this is looking at gene expression array data in those same human PBMCs, this is looking what happens when you co-incubate PBMCs with LPS. You see upregulation of all the inflammatory pathways, exactly what you'd expect. If you take human cells, incubate them with the inulin adjuvant, you're now actually, in green, is downregulation. So this is TNF-alpha, interleukin-1. We're actually downregulating all the inflammatory genes in the cells that are taking up the inulin particles. Now that suggests that, in fact, inflammation's got nothing to do with adaptive immunity. And you can induce adaptive immunity without inflammation, which really immunologists are still finding hard to digest, but that's what the data shows. Suffice to say, there are lots of pathways involved in this that we're dissecting out, but largely it appears to work by improving antigen presentation. Co-stimulatory factors go up. It increases chemotaxis to the injection site. It differentiates monocytes into dendritic cells, but it doesn't induce any inflammation, and this really is what sets it apart. So we believe that it tells us something about the immunology that previously wasn't appreciated, that inflammation isn't necessary to make a good adaptive immune response. And in fact, what we now think is that in fact inflammation inhibits an adaptive immune response, and the reason we get such great protection is the very fact that we're not inducing inflammation, so it allows a full-fledged memory response. And so, in fact, we're working our way through, but we think we've got solutions to a lot of problems, but obviously there's a lot of work to be done. But essentially what we think is happening is immunology got it wrong. They thought the more inflammatory, the better the memory response. What we're showing is the more softly and the less inflammatory the environment in which you induce an adaptive immune response, the better and the longer lasting the adaptive immune response is. So as I say, softly, softly, catchy monkey. So we need to acknowledge all the people who've done this work, an enormous number of collaborators, but particularly the NIH, which have funded us for the last 10 years. And yesterday I just got the nice news that they've given us funding for another 5 years, and the Bill and Melinda Gates Foundation, who funded us to do some polio work. So thank you very much. And we're pretty well right on time. So time for just a few quick questions and then we'll break for coffee.
Fantastic development. Can you tell us a little bit more about the molecular weights of the immunants that you use and how important it is to get the right molecular weight?
Probably a branching issue with inulin as well. Is it the right degree of branching that you have to have? And what you've got is not a crystallization, but more of a precipitation. This doesn't look like a crystal to me. If you put it in an X-ray if you've done X-ray crystallography on it, in which case, let's know about that.
Good question. So the answer is yes, the polymer length is absolutely critical because short polymers don't crystallize, and I reinforce that term because it truly is crystallization. Polymers do crystallize, but when they crystallize, they don't look like salt crystals. They don't have sharp edges. But if you do X-ray diffraction, or recently we've done some studies with the Australian synchrotron, and we can clearly show that these are crystal structures where the polymers actually form into helices, and then the helices all line up and form a beautiful crystal structure, just like you would do with, say, a protein crystal. So the fact they don't have sharp edges doesn't mean they're not crystalline. In terms of branching, again, you can't have branching because branching stops crystallization. So you need to have these linear, non-branched inulin chains. So I guess a lot of the intellectual property that we've developed over the last 10, 15 years is working out exactly what parameters in terms of inulin sources, the length of the polymers, the fact they're not branched, and how to get them to crystallise in this way that gives them the adjuvant activity. If we just heat them up and dissolve the crystals, they completely lose all immunological activity, and you can use them now for renal function testing again. So it's a beautifully reversible process, and similarly we believe that's why we don't get toxicity, because in fact in the body these are going to get hydrolyzed, become soluble, and then they just go out through the kidney, because inulin itself has no biological activity in its soluble form. So it's
iodine staining starch also has a spiral structure. If you do the reason iodine stains starch, which is polyglucose, that also gives you is a result of a spiral structure. You might want to have a look at crystalline starch as well.
No, we've looked at a lot of polysaccharides, because obviously the beta-glucans have been known to have adjuvant activity for over 20 years, and that's polyglucose, beta-glucan. The interesting thing about all the other polysaccharides we studied is they're all highly pro-inflammatory because they're binding to dectin-1 and other inflammatory receptors on dendritic cells. So inulin crystals were the only polysaccharide we could find that was non-inflammatory and yet had beautiful adjuvant activity. Again, saying it's something very special, and we're trying to understand what it is and how those particles are interacting with the immune system to understand why that is. Why is it not inflammatory, unlike polyglucose?
A quick question. The dendritic cells are key, and I was wondering if you look at PBMCs, but did you ever look at the DCs to see increased either maturation? Does it have a direct effect? Does it also affect the kinetics of the response when you did it in vivo as far as looking at primary versus secondary?
Yeah, so great question. The answer is yeah, we looked, and it appears to take an immature monocyte lineage and take it to an immature DC. It's certainly unlike LPS. You need an inflammatory stimulus typically to mature an immature DC, and it doesn't do that. So if we take immature DCs, add the inulin, we see absolutely nothing happen. If we take monocytes and add inulin, we see, upregulation of CD11C, MHC class 2, CD86. So they look like they're turning into an immature DC. That's in vitro. In vivo, we think that they actually mature all the way despite the lack of inflammatory stimulus. But again, it's hard to do those studies in vivo to understand that.
In terms of T-cell differentiation with no inflammation, do you see regulatory T-cell generated, Th17 cells?
No, so we've been looking at that more recently. So the question is, do we see Tregs generated? And we think to induce a Treg, you need inflammation. And so again, one of the reasons we think we get such beautiful T-cell memory is if we're not inducing Tregs, we're not getting pushback. So we think if you use an inflammatory stimulus, a lot of your T cells go into Tregs, and that then pushes back and stops further development of T cell memory. So when we looked at FOXP3, we don't see really upregulation with the inulin adjuvant. So we don't appear to be inducing a Treg population. It really is I guess a central memory. So if you talk about T cells as effector memory or central memory, what it's clearly inducing is a central memory T cell rather than an effector memory. Although that said, we see a lot of gamma interferon produced by those T cells on a memory, recall response in vitro. So again, we're trying to understand that, given that there's no inflammation at the time of immunisation, and I think Jean-Pierre Schierling, who's here, did some sheep studies and demonstrated beautifully there's no inflammatory cytokine even if you sample the draining lymph node at the site of inoculation. So it really is true. There is no induction of inflammation at the injection site of the inulin adjuvant, and yet you get these lovely gamma interferon producing T cells on a recall response.